Thanks very much. I'm happy to be here. Uh, so today I'm going to be talking about morphological and molecular analyses that reveal COTS brain novelties. Although I'm not necessarily going to get into that early on, I will get into that a little bit later. So I lead a research group called the Molecular Communication Research Group at USC, University of Sunshine Coast, and we kind of focus on endogenous communication signals within invertebrates, and most of them are aquatic invertebrates. So thinking about hormones and how that influences the behavior of invertebrates, and also exogenous signals, so things that they release into their environment, which influences many different things. So I'll jump onto that exogenous or semiochemical topic, and I'm sorry if I'm oversimplifying things here, but how I describe it is, all animals have developed various ways of sensing and responding to the environment. We, of course, primarily use vision and hearing, but if you're, say, a fish or a starfish or a squid or a snail or a sponge, they have very limited amounts of eyesight and also hearing, but they make up for that by having an excellent sense of smell or chemical communication, which they can and do use in many, many critical life situations, from knowing where to settle, to finding food, and recognizing when predators are nearby, and also recognizing the difference between a conspecific, so something that they need for mating purposes. So with that, over the last maybe 10 years with the developments in technology, we've been increasingly been able to kind of eavesdrop and understand this chemical communication system. So we may not see it, and there's a good reason for that, because it is their own dialect, it is their own kind of communication system. But with new technologies, we can understand the chemistry, and a lot of these molecules are small molecules, secondary metabolites, and what we discovered over 20 years ago is that peptides and proteins are often used as chemical signaling devices for aquatic organisms. So these molecules are saying something different, and they might be quite unique, and often they are unique to a particular species, and that's the basis of a pheromone. It might be saying something like, hey, danger here, or it's time to spawn, or something like, come over here, I want to mate with you, something like that. This was based on research that I was lucky to be involved with almost 20 years ago now at the University of Texas, where we identified the first peptide or protein pheromone system. And this was in sea slugs. So why sea slugs? Because we knew a lot about their behavior at that point, at least within the molluscan world. So we used a process of bioassay guided identification and purification to identify what was attracting conspecifics to themselves. And we realized that it was a group of proteins, a cocktail of proteins, and we identified them. They were novel, and we called them attractin, enticin, temptin, and seductin, which was a great name because that's exactly what they do. They attracted them and, pulled these, these are animals that live in, same as crown thorn starfish, large spaces, but they had to come together at very defined times of the year. And we could even make recombinants of these and show it in behavior assays. This is just a very basic T-maze assay where we could put very small amounts, picomolar concentrations of one of the pheromones, attractin, and we could see that it was attracted to the conspecific. All right, so from this point on, in the last 10 years, I've kind of developed a group, and we kind of utilize the breadth of omics analysis from the genome. We need to understand the genome if we want to know all the proteins and also somewhat the metabolome. And if we can't get a genome, then we'll have transcriptome, so all of the RNAs that are produced. We now work on a large group of different types of, mostly aquatic, but there is a land snail there, but mostly aquatic animals, looking at chemical communication and linking in with various institutions, including AIMS and funding bodies. And generally the questions are related to biocontrol, aquaculture, ecology, and health. But I do want to focus here on the crown of thorns starfish and how we might be able to utilize this information. Several years ago, I was lucky to, or our group was lucky to integrate within the Genome Project. But secondarily, we were also working with AIMS in relation to excretory, secretory molecules from the crown of thorns starfish, which linked into the Genome Project. We needed the genome to kind of have this database to understand the molecules, and in particular the proteins that are released by the crown of thorns starfish. We looked at it in the context of aggregation, so recognizing that, well, anecdotally in the field, they do come together, but replicating in a Y maze, and that's what this figure represents, and it's a heat map showing there is increased activity and movement towards aggregation. Therefore, some sort of molecules are released from these aggregating starfish, and also in the context of stress. So when you expose them to a giant triton, which is releasing its own odorous molecules, do they run away? And well, yes, they do run away. So it's kind of an inverted Y maze assay. So what were the actual molecules that the COTS were releasing? And we discovered there were a number of unique proteins, and you might think of them as kind of those words that are telling the organism to do something. And some of them, had names already. So people had discovered them in other species, not so much the crown of thorns starfish. And it was interesting in the aggregation that you do see some of the toxins. So toxins, not just in the spines, but they're also being released into the water. And also in stress, they had different types of proteins. And there were a large number of novel proteins. So these are the ones we kind of think are species-specific chemical communication devices. But there was another 102 proteins besides these. And so that's about 108 proteins, which might be the vocabulary of a crown of thorns starfish releasing molecules into the environment. We think that, well, they might be involved in predator defense, some sort of zone of protection, like the planty toxins, which is a toxin, but some of them, or quite a lot of them, might be involved in pheromone communication. At USC and in conjunction with research at Okinawa Institute of Science and Technology, in pheromone research, we're only as good as the behavior assays that we've got available. And I thank PhD student Adam Hilberg for a lot of these developments, and also our collaboration with Russell Wyeth, who's very much in the field of aquatic animal behavior. This one here we call the box assay, in which we give them choices, and you see this guy here is attaching to this box here. We can put stimulants in there. They can't touch them, they can't feel them, they can't see them, but they can smell them. So doing bioassay guided fractionation of what they're releasing, we can put into these boxes. A relatively simple assay. A bit more high throughput is just what we call a box assay, where we can look at them underneath and just look at increase in activity. And well, this example here is actually looking at acropora mucus. So realize more and more how important acropora is, especially from the previous talks. And they are very, very excited about this acropora mucus, not so much the actual acropora itself, it's what it's releasing. And we ultimately have to move into something that's more behavioral and more movement-wise. So Y-maze assays are our kind of bread and butter towards identifying that. So this is a relatively small Y-maze assay compared to what's available at AIMS. All right, so this leads me into more the novelties in the brains of, or the nervous system of the crown of thorns starfish, where several years ago, I kind of thought that electrophysiology might be a really good way of understanding sensory responses, because it's been done for 40, 50 years in insects and also in mammals. And I thought it would be relatively easy with crown of thorns starfish. So this links into the neurons, and we've give them some sort of stimulus, some sort of smell, and that would provide something like an action potential like this. And then it would say, oh, well, yes, they can definitely detect it. However, I realized quite quickly that there was a large gap in our understanding of the neural system of starfish, not just crown of thorns starfish. And that led to a whole area of research, PhD research, where we discovered the neuropeptidome, all of the neurohormones that are produced by the starfish, also the small molecule neurotransmitters. And we even went into some functional analysis on a particular peptide called relaxin-like gonad stimulating peptide. So for those of you who are not really aware of the neural system of the crown of thorn starfish, here's a crown of thorn starfish. I've outlaid the neural system, which involves a ring, but also radial nerve cords, which goes down each of the many arms. So we were particularly interested in the radial nerve cord. We had a look at it under the microscope and under the scanning electron microscope, and we do see these, what we call neural bulbs protruding. We didn't really think much of it at the time because there wasn't much known about this radial nerve. But when we looked at the juveniles, we didn't see these radial nerve bulbs. And when we looked at the sister species, the Acanthuster brevispinus, this SEM showed that it was completely clear. So these bulbs were not present. So we got even more interested in these. This is a tissue section, cross section, showing again radial bulbs protruding from the ectoneural region, not present in Acanthuster brevispinus. And even closer, we look at transmission electron microscope, and we can see that a lot of these bulbs are made up of mucus, but we also were lucky enough to see this transition stage of secretory vesicles where it's actually excreting some sort of substance. So we were able to do a proteomic analysis, and we found that a few of these proteins were actually released into the water. So these are the candidate pheromones. Just quickly, I'm going to follow up on one protein, the RGP, the relaxin gonad peptide. We made a protein, a recombinant protein of this. That's what's shown just here. We put it into an in vivo assay where we got the tubules, the gonads of a female crown of thorn starfish, and we could show that it induced germinal vesicle breakdown. So this wasn't random. We already knew that someone had researched them on Asteris rubens and shown that it had something to do with reproduction. But it seems that this is a key molecule as a hormone to induce germinal vesicle breakdown and probably spawning. Fortuitously, we were working on a project for OS for Innovation, looking at reproduction bottlenecks in another echinoderms, the sea cucumbers. And we did the same thing, made a recombinant for a sea cucumber relaxin-like gonad peptide. When we put it into the sea cucumbers, what they do is this little head waving, and that's part of their reproductive behavior. And ultimately, they gets to it, undergoes the spawning. This is a male who's spawning. So we think it should happen with crown of thorn starfish. We haven't tested the recombinant on the crown of thorn starfish just yet. All right, getting back to the pheromone story, from the 108 proteins using in silico and in vivo analysis, we've narrowed that 108 proteins down to a more manageable lot, and that's the list here. When we look at the expression of these genes within the different tissues, so this represents an expression heat map of different tissues, red meaning there's lots of it, we see that the spine is the main source of these candidate pheromones. What does that mean? Well, we hope that maybe with more research, we can identify some of these pheromones, these attractant pheromones that might be used on the reef as some sort of releaser that might pull the crown of thorns starfish out of those cryptic regions that Morgan spoke about. And that might be really important to understand what is the true density of these guys. And, well, maybe we could put them into some sort of trap as well. So I just want to finish off by just talking about another project, thinking about something that's different compared to what we've worked with in the past. And this relates to a parasite. It's parasite's called schistosome. It kills around 200,000 people per year, mostly in third world countries. It infects them and kills them over a period of time. So we linked into this through an ARC discovery project, recognizing that there were aquatic stages of this parasite, the Immurocydia and the Cercaria, and they had the host of a snail and obviously the human. We used a similar workflow. So just thinking about it, they've worked on this disease for about 50 years and they haven't come up with a vaccine for 50 years. So this was thinking about something novel and thinking about something different. We looked at what the snail was releasing. Bioassay guided analysis identified a peptide, which is completely novel to this particular snail. And what we can do now is, well, we can put it into and give it to this Miracidia, the parasite, and it's, the parasite thinks the snail's there. So we're not thinking too far out the box as far as the crown of thorns starfish. We do know that we can identify the pheromones. It's really just a matter of what we can do with them. And hopefully we can cure this disease, schistosome. Just finally, I just want to thank my group, molecular communication group at USC, which includes bioinformaticians, Min Jiao, Tianfang Wang, proteomics, a few molecular biologists, and a number of PhD students, as well as a variety of people, including Maria, who's helped enormously with the histology and physiology of the radial nerve cords. Thank you.